This lesson is going to be our first of several about the structure of DNA and how we determine the structure of DNA. I'm kind of excited because it's very interesting, the story of how we discovered the structure, but also understanding how it works. It's kind of one of those things where you find that it's ingenious. You know, nature came up with this fantastic way of transmitting information and determining all these things. So I hope you find it as exciting as I do. We're going to start way back in 1869. We're going to be talking about some different individual people. The first is Friedrich Maisel. In 1869, he was the first to isolate DNA. We didn't call it that then. We called it just a nucleic acid. But he discovered in the nucleus of every cell that there were these substances that were acids. And so he called them nucleic acids after the nucleus of the cell. So he was the first to isolate DNA. Later, this fellow Frederick Griffith in 1928, he did a series of experiments that helped us to understand that there was an important molecule of some kind that was passing on information that was going to be the key to understanding how life works. So we're going to talk a little bit about Frederick Griffith's experiments. He worked on pneumonia bacteria. So you may have heard of pneumonia before. It makes you really sick. Some people get it. It kind of infects in your lungs, and it makes you really sick. So he was studying pneumonia bacteria and maybe trying to work on a cure. This is a microscope picture of some pneumonia bacteria here. You might notice that the surface looks fuzzy. We're going to talk about that in just a minute. So as he's working on these pneumonia bacteria, he isolates two strains. The first is called S strain, S for smooth, and the S strain he found to be pathogenic. Pathogenic means it can cause illness, and it can make you sick, and it can kill you even. Okay, so the S strain was the one that made you sick. The R strain was the other strain that he isolated. It had a rough surface, like the one you just saw. And this strain did not make anyone sick. It was non-pathogenic. Now here's his experiment, very ingenious experiment. He injected the R strain into mice, and of course they survived. It didn't make them sick. He also had separate mice he injected the S strain into, and they got pneumonia and died. Then he heated the S strain. Okay, this is the one that makes mice sick. He heated it up and injected the cooked S strain into mice, and they survived. So he found that the cooking killed the bacteria. Okay, so here's a summary of that. The S strain kills the mice. The R strain does not. And if you heat up the S strain, they survive because the S strain, the bacteria are killed by the cooking. Next, and this is the ingenious part, he mixed cooked S strain bacteria with living R strain. Remember, the S strain kills people, but it's cooked, so it doesn't. And he mixed that together with living R strain bacteria, which do not kill anyone. But he mixed these two different bacteria that should not have hurt anyone. They were non-pathogenic, but when he injected those into the mice, they died, and this was a surprise to him. Okay, and then after the mice died, he took their blood and looked at it under the microscope, and he saw live S strain bacteria. So somehow, the R strain bacteria were being converted somehow into S strain bacteria. The living R's had become S's somehow. Very strange. So he determined that somehow some information was passed from the dead S strain to the living R strain, which transformed them into S strain bacteria. And later we find this information that was passed along was DNA. Later in the semester, we'll talk a little bit about exactly how bacteria can transform each other like that. But he discovered that there was something being passed from one bacteria to another, and this was very exciting. This was going to be the key to life. Now, many years passed, and now we're in the 1950s. Around this time, it became very clear that DNA was going to be important. So many scientists were searching to discover its structure, to understand how it worked. It was pretty much well understood that whoever figured it out was going to get the Nobel Prize, and that's, of course, what scientists want. It's like the Oscars for scientists. They knew some things. They knew DNA was composed of something called a phosphate. They knew that it had a sugar in there, and you know what the structure of a sugar is. It's a ring. And then it had 4 different nucleotide bases. It had these nucleotides that made it up. They were adenine, thymine, guanine, and cytosine. And we usually abbreviate them A, T, G, and C. Now, the other thing that we knew was due to this fellow, Erwin Chargaff. He discovered that when you look at DNA from any organism, the amount of adenine was always the same as the amount of thymine, and the amount of cytosine was always the same as the amount of guanine. And this became known as Chargaff's rules. So it was pretty well understood that there was going to be some reason why A and T were always the same and C and G were always the same. It was kind of a curiosity. We also had some information about the structure of DNA from this scientist, Rosalind Franklin. Rosalind Franklin was doing some techniques using X-rays, and she found she could crystallize the DNA and take X-ray images of the crystallized DNA. And this is an example of one of her pictures. I know it probably doesn't mean anything to you, except it looks kind of cool. But this picture is very famous because by looking at this picture, we could find that DNA had a repeating pattern, and it even gives you an idea somewhat of the structure by looking at these images. So her finding was extremely important. But none of those people that we talked about were the ones that actually discovered the structure of DNA. The actual structure of DNA was discovered by these 2 men, James Watson, who's an American, and Francis Crick, who was English. And together, what they did was they put together information from all these different places, and they worked out the structure by just thinking it through, coming up with the idea. And together with the scientist whose lab they worked in, Morris Wilkins, they all received the Nobel Prize for finding the structure of DNA. Okay, so they took information from different scientists, and they found the structure was something called a double helix, which is a spiral shape. Think of snail shell or something like that. And it's kind of like a twisted ladder. The phosphates and the sugars are the sides of the ladder, and the bases are paired up in the middle according to Chargaff's rules. A with T, C with G. So this is kind of like what it looks like. If we color code our bases, A and T, notice here, A and T are coming together, and they're always both there, and that's why you have the same amount of them. And then C and G, same thing. Cytosine and guanine, they always pair up, and that's why you always have the same amounts of those. The sides here are made up of sugars and phosphates. And that's the basic structure of DNA. Here's a more detailed picture. Again, we have the twisted ladder. If you straighten out the ladder, it looks something like this. And then you can see, again, adenine with thymine here, guanine with cytosine here. And then in this diagram, you can see the alternating sugar and phosphate groups here. The sugar is represented here by a little like 5-sided ring, and then the phosphate's just that little yellow circle. The whole thing is actually 2 strands, one going this way, one going this way, and it's held together here in the center by hydrogen bonds, and you know what those are. So not only are hydrogen bonds extremely important with properties of water, but now you're finding that hydrogen bonds are crucial to our structure of DNA. And we'll talk more later about why it is that's so cool that there's hydrogen bonds in there. All right, that's it for these notes, and we'll see you on Tuesday.